Good morning, everyone. I would like first to thank the organizer for giving me the opportunity to present my work. I'm defending my thesis tomorrow, so I'm very glad to present to you the main result that I obtained during those 4 years here at the ICM in a team of Séverine Boilet, directed by Delphine Ball, and I'm working on propagation and toxicity, as Pascal said, of SOD1, sorry. So as you may know, 30% of the ALS genes encodes for proteins who have prion-like domains or prion-like properties, and among them, I focus my thesis on superoxide dismutase 1, SOD1. As you know, more than 200 mutations have been linked to ALS on these genes since 1993, and those mutations may lead to aggregation or accumulation in postmortem tissues of patients, and also may lead to aberrant protein conformation that can lead to an acquirement of prion-like properties. And taking those prion-like properties of SOD1, we can think that maybe it can be secreted and may be implicated in the propagation of SOD1. That's why we think that maybe by modulating the propagation of SOD1, it could be of therapeutical interest to limit ALS progression. So concerning SOD1 secretion, thanks to several studies that have been very useful to show that SOD1 can be secreted through different models. We know now that SOD1 can be secreted, but those studies were always in overexpressing models and non-neuronal models. That's why in my thesis we ask about this question of propagation and toxicity, but at a steady state level using iPS-derived motor neurons from ALS patients. So to do so, I'm using iPS cells that we have in a lab, and I'm using 4 different iPS cell lines mutated into SOD1, as you can see here, and I'm comparing them to non-related control and isogenic clones generated by CRISPR-Cas9. I'm then differentiating them in mature motor neurons that we can obtain in 35 days. And then to follow the propagation of SOD1 in those models, I'm using the C4F6 antibody. It's an antibody that has been developed in the lab of Jean-Pierre Julien. It's specific for the misfolded forms of SOD1, and it's been validated in several models. And so firstly, we wanted to know if our motor neurons can if in our motor neuron we can observe the inclusion that is found in the postmortem tissues of ALS patients. So by performing immunofluorescences using this antibody, as you can see here in the mutant motor neurons compared to the controls, there's those green spots that show misfolded SOD1 inclusions. And in the control, you can see that there are just very few of them. They are not very intense and not that big. And when we use a native antibody just to see the normal expression of SOD1, there's no difference in the control and the mutant. So we decided to quantify the number of those inclusions, and now you can see here on the graph on the left, this is the number of the C4F6 positive spot per cell, and as you can see by comparing, there is a clear and significant increase in all of the mutant cell lines compared to control. And interestingly also, when we look at the volume of each of these spots, we saw that they were bigger, in the mutant they were bigger compared to the control. So firstly, we thought that in our model, there is misfolded SOD1 protein that accumulates in mutant SOD1 motor neurons. So now that we know that we have those inclusions, we wanted to know whether they can be secreted. So we focus on different pathways, but I'm going to present here just a very interesting one that was already presented here yesterday, by Philippe Gosset. It's the MAPS pathway that stands for misfolding associated protein secretion pathway. It's a pathway that is specific for the misfolded protein secretion. Just quickly, it's initiated by USP19, a deubiquitinase that must be fixed on the ER to be active in this pathway. It deubiquitinates misfolded protein, and then those misfolded protein are sent towards DNHC5, a chaperone. They form complexes together, and those complexes stay together until secretion. So firstly, to see if the MAPS pathway can be involved in those motor neurons, we looked for those complexes. And this is what we observed by performing confocal imaging. As you can see, there's some green spots that are misfolded SOD1 accumulation, and they are surrounded by red labeling showing DNHC5. And so we decided to quantify this colocalization, and as you can see here on this graph, this is a number of the colocalization between misfolded SOD1 and DNHC5, and you can see that all of the mutant cell lines, there's a clear and significant increase in the number of those colocalization per cell compared to the control. So firstly, concerning this pathway, we showed that DNHC5 can form complexes with misfolded SOD1. But those results were in a soma, so we also wanted to know whether we can find those results in the axon and in the rest of the motor neurons. So to do so, we use microfluidic devices by putting just on one side mutant motor neurons and nothing on the other side to just look at the neurite network. And as you can see here in the neurite and also in a growth cone, you can see some green spots, so showing misfolded SOD1 inclusions, and some of them colocalized with DNHC5, that may leading us to suggest that there might be a transfer of those complexes through synapses. And that's why at the moment we are performing experiments where we add on the other chamber either wild-type motor neurons or muscle cells to see if there is an uptake by those cells. To also more understand how the MAPS pathway can be involved in the SOD1 secretion, we wanted to modulate USP19 because, as I remind you, this pathway is initiated by USP19. And so when we downregulated USP19 efficiently with lentiviral vector, as you can see here, and when we looked at the number of the colocalization between misfolded SOD1 and DNHC5, when we compare the sRNA control and the sRNA number 2 that efficiently decrease USP19, you can see that there is significant decrease from some clones of the number of the colocalization per cells. Even if for some clones it's not significant, there's still a trend to decrease. So we show that USP19 downregulation may impact the formation of the colocalization. And to know more about how downregulation of USP19 may also impact the rest of the motor neurons' behavior, we perform RNA sequencing. Here you can see the result of the transcriptome comparison for the mutant cell lines when we compare sRNA control and sRNA that downregulate USP19. And you can see firstly on this Volcano plot that a lot of genes are deregulated, and also on this GSEA that there is 7 pathways that are deregulated, and one of them is a protein secretion pathway, so very interesting in our case. So we looked more on for this pathway, and among the genes of this pathway, there is 39 that are clearly downregulated when we downregulate USP19. And when we look at for those 39 genes at the protein level, performing string analysis, we saw that 30 of the 39 downregulated genes interact together at the protein level, and some of them form hubs. So we wanted to know more what were the role of those hubs. And as you can see here, I'm not going to enter in the details, but some of them are really involved in the early exosome, early endosome structure, late endosomes, phagophore, autophagosome, the communication between endoplasmic reticulum and Golgi apparatus. And so the downregulation of those genes may really mean that there could be a real impact of USP19 downregulation on vesicle trafficking and a disturb of protein secretion. That could be not very good for motor neuron in the end. So to conclude briefly, we showed that misfolded SOD1 can accumulate in mutant motor neurons. We showed that SOD1 can be secreted by motor neurons. I didn't have time to show you, but we showed that the ER-Golgi pathway might not be involved in this secretion. Concerning the exosome pathway, the results are not that clear for the moment, but we're still looking for it. But the MAPS pathway might be involved because of we found DNHC5 and misfolded SOD1 colocalization. I did not have time to show you neither, but USP19 upregulation did not show any upgrade in the number of colocalization. However, concerning downregulation, USP19 downregulation, a good part of this is that we diminish the number of complexes between DNHC5 and misfolded SOD1. That might lead to less SOD1 secretion and less maybe diminish the ALS progression. But however, as I showed you, there's a deregulation of the protein secretion pathway that could really be toxic in the end for motor neurons, also because it's very important at the synapse level. That's why, in the perspective of this project, we still want to see the MAPS pathway as a therapeutical option, because there is mechanism that I will not really be in a detail about that, but USP19 can auto-inhibit its Either it's the ubiquitinate activity or inhibites its anchoring at the ER, and thus, in the end, by using those mechanisms of autoinhibition, it could still be maybe possible to modulate the MAPS pathway to try to limit the propagation of misfolded SOD1, but also maybe misfolded protein that have prion-like properties like TDP43 or FUS to, in the end, maybe try to extend the lifespan of the patients. And I would like to thank you all for your attention. Thank you to all the ICM teams, our collaborators in France, and all the platforms here that help us during this project. Thank you.
Thank you, Coline, for this nice talk. Do you have any questions? No? I have one question about USP19.
Yes.
We know that when you overexpress USP19 in your cells, in egg cells, you strongly stimulate the release of TDP43. Did you try, when you overexpressed USP19, to see if SOD is released or not?
I didn't have time to show you, but we tried to overexpress USP19. Concerning the SOD1 secretion, we did not see any difference. And concerning the colocalization between DNAG5 and misfolded SOD1, there were no difference neither. But we hypothesized that it's because maybe USP19, even if we overexpress it, is not fixed on the ER. Mm-hmm. So we are at the moment performing some experiments to do some fractioning of the cell lysates to see if USP19 is really fixed on the ER and can be implicated in the MAPS pathway. Because if not, maybe it's the explanation why there is no differences.